you've shown up, and I made you co-host so you can like do things. I don't know what you can do as a co-host, but you can probably do certain things. But I just said that co-host is a kind of salmon, but you just take off the S and the T. Co-ho. So other people are still coming in, and I'm still using this. I use this waiting room thing because since we put this out on Facebook, we have a possibility of being bombed. And Sue was in a meeting the other day where they were bombing. They came in and started saying all kinds of vile things, and the person who was running it was not quite prepared, and they had to do quite a bit of mouse clicking and things. I think if it happens for us, I would just like hit the button and end it all. So a big nuclear holocaust would happen. Oh, not that kind of end at all. I mean, you know, end the in the session. So some good news. We've been asked, Sebastian, Augie, and John, Genevieve, and I have been asked to present at the ASP meeting this year how we actually presented and taught field parasitology online last summer. So we're going to do that at the ASP meetings this summer. So I think we have to send in an abstract soon. So we'll see about that. If I guess if they really want us, they can write our abstract for us, perhaps. Okay, more people coming. I'm not going to. Here they come. So I'll have Sebastian write the abstract, and Augie can edit it, and John can then submit it, and then I'll just sit around. Yeah, that's not true. I'll just go ahead and do it. Okay, so what else? Any other announcements? This is the last seminar of the semester of the second year of COVID, of spring semester, and we will resume again in August, probably like August 20th, the week of August 20th in there somewhere. We'll be teaching at Cedar Point again this summer. So we will have, Augie will be the TA, Kevin will be a student, so he'll be learning how to do the TA stuff, and I'll be there in my usual supporting role to those guys. So let's get going. Are you about ready, Sebastian? And I think you have the ability to take control of our screens.
Yeah, I think
just like the old Twilight Zone and Rod Serling. Don't try to adjust the controls because the screen has been taken over by you. So Sebastian finished his PhD in 2020 in the Manter lab in the School of Biological Sciences, and now he's running an NGO in Medellin, based out of Medellin, Colombia. He's also still associated as an associate of the Manter Laboratory. And so we've been working together with Augie, with Batsaikon and other collaborators, Gabor especially, to look at some of the data that we collected in Mongolia over those 4 or 5 years that we did collecting over there. So Sebastian agreed to go ahead and do a presentation today, which we really appreciate very much. So thank you very much, Sebastian. And he's going to speak about the meta-community structure of Mongolian mammals, current and historical drivers of biodiversity organization in the land of Chinggis Khan. So take it away, Sebastian. Thank you so much for being here today. Go ahead.
Thanks, Scott, for the introduction, and thanks everybody for being here. I have a couple of disclaimers before I begin. Well, first, you already saw it with the title. This is not about parasites, but it's using the data gathered by the Mongolian Vertebrate Parasite Project, and eventually we hope to include parasite data to better understand these parasites, and as well as to describe these patterns for parasites. Second, this is a very preliminary work. I am still wrapping my head around the methods and how to analyze these data sets. So if I'm not clear with something, or if you think, or if you have ideas about how to do this kind of work better, please interrupt me and let's discuss it. I want this seminar to be basically a conversation about what we can do with these data sets. And finally, all the cool pictures that are in the presentation are from Scott and the field team that did the work of going there and collecting the mammals. None is mine.
So Sebastian, we're seeing we're seeing the editor mode. If you can go into the presentation mode, then we'll just get one screen. We can see both screens. We could prepare for what you're going to show us, but it's probably better just to have you show us.
A surprise.
Yes, yeah, we wouldn't. We prepared for each surprise there.
Let me see, how can I fix that?
I don't remember.
Here.
That's it. Then I think if you just hit that big thing on the bottom, it'll go full screen. Yeah. Didn't work?
No.
Oh, that's weird.
Wait. I'm sorry, give me a second.
It's probably because you're closer to the equator. You know, when you're closer to the equator, things happen differently because the Coriolis effect is stronger down there because it's going the other direction just on the other side of the equator. What are you guys at, about 30 degrees? No, 20?
7 degrees.
7
north.
degrees. At Medellin?
Yeah.
Okay. 7 degrees. So that's why you're really close.
Okay, sorry, give me a second. This is going slow, but almost there.
That always happens. If anybody comes in who I don't see, let me know, because sometimes I miss people and they just sit there in the waiting room for like an hour having some hot dogs and things.
And there, it's working.
Yay, works great. Thank you.
Okay, perfect.
Okay, I'll mute myself. Sorry.
Did I pass the? There, can you see the next slide?
Yes.
Okay, perfect. Thanks. So we know that understanding the mechanisms determining or influencing the composition of species communities at all scales, from global to local scales, it's a key question in not only in ecology but in biodiversity science. And it's not a simple question. It's a very complex question because it involves several scales of analysis, both taxonomical and spatial scales. Especially they can go from local species pools to regional and global species, and the factors that from like short term, such as interactions, to long term, such as speciation and extinction, that drives the composition of these species pools. And this is a very important question, not only for basic science, but it's key to understand what is happening now with the global environmental change we are facing. We need to understand what mechanisms influences species assembly rules and how we can predict and manage the effects we're having over the biosphere. Ultimately, not only influencing species extinction and composition, but things like host switching and host switching and recomposition of host parasite assemblages. And a very useful framework for starting to tackle these questions is the meta-community framework, which sees the composition of a species like a set of local communities embedded in a larger region where species are shared through dispersal. So this is a visual model of a meta community. We have several local communities with its own populations, and a meta community will be the set of those local communities through a region. Meta communities are dynamic. They can change species through dispersal. Species can go extinct, or new species can come in through dispersal or through speciation. So this gives us a framework to start understanding the local and regional process affecting the composition of these local communities and the result of this for the entire regional species pool. And if we assess meta communities composition through environmental gradients, we can also start assessing or thinking about the mechanisms that of how these gradients influence species composition through time and space. And so thinking about these ideas, we came up with the idea of using the data set from the Mongolian vertebrate parasite project, which was a sampling scheme through Mongolia that lasted more than 10 years and sampled around 30 localities through the country for vertebrates and their parasites as a very good data set that, with which we can start applying these ideas and start thinking about what are the drivers and the mechanism, well, the patterns first, and the mechanism behind the patterns of a species composition through these landscapes. The Mongolian Vertebrate Project is a large collaborative project, including the National University of Mongolia, the University of Nebraska, University of Kansas, University of New Mexico, Birmingham Young, and the National Museum of Natural History of France, with a large sampling and a large field effort through the entire landscapes. So the idea here for this talk, at least, is to use this data set to explore how the diversity of small terrestrial mammals in Mongolia is organized through the diverse habitats sampled by the project. So basically, the first goal is to assess the patterns of organization of a species through the landscape using a meta-community ecology framework. And then the second part, it's after showing you what we have found, what patterns we have found, discuss the potential mechanisms behind this pattern and what we can learn about the effects or the different mechanisms driving a species composition of local communities and at the end in regional communities. So first, a little bit about the theoretical framework of meta-communities. Basically, we analyze communities as, again, as I said, as sets of local communities and through landscape or through an environmental gradient. It uses this way of depicting or thinking about a species occurring through the geographical space. We have here the sites, in this case, for this example, that it's from Manu National Park in Peru, rodents. We have little bands of elevation. Yeah.
You're still only on the species ecological assembly rules slide.
Oh, shoot. No, I have skipped like
I skipped a couple slides. So
a couple of slides, yeah.
Go back to the meta community one there. Oh yeah, this one. So
is it moving there?
Okay, yeah, it's moving. Meta community. So okay, yeah, that one. So just quickly run through that one and then say next slide, and if it goes, then tell us which slide you're looking at just to make sure it's going next time, because it made good sense, but then when you started to say something else, and it kind of disintegrated into us wondering what was
well,
happening.
thanks
So,
for letting me know.
sure.
So yeah, basically here, this is a graphical model of how we can think about meta communities. First, we have local populations that are part of local communities, and a set of local communities within. Can you see the mouse there or not?
Yes, we can.
Okay, perfect. I think now it's working. I don't know what happened. And then we have all these local communities through a landscape composing or making the a meta community. Here I talked about the Mongolian vertebrate parasite project. Here we have the collection localities spanning a large geographical area as well as a diverse set of habitats. These are the collaborators of the Mongolian vertebrate parasite project. And well, yeah, I already talked about the goals, and I was here talking about the theoretical framework of meta communities, in which basically we can think of a meta community as a set of sites here on the y-axis and a set of species on the x-axis, and we can assess how species are distributed through the sites we have, and this gives us a pattern of how species are how species change through a space. We don't necessarily need to know which is the environmental gradient here. For this example, which is for an elevation gradient in Peru, we have the elevation, and we can see that elevation is having a large effect on the composition of a species at each site, but we don't necessarily need to know the environmental gradient there. We only have the sites by species, and basically what we can do with the meta community framework is assess what's the pattern of organization of these species and how species change or turn over from place to place. And the pattern of change can be attributed to several idealized structures and ideas about the mechanisms that drive a species composition, ideas that go back to Clement about the community unity hypothesis, which he said that communities were like units where species had evolved together and were together through space. So in that case, we will see in a pattern that we said see sets of species changing from place to place. But of course, we know this idealized pattern, it's not very prevalent in nature. Then we have, for example, Gleason, which said that species show idiosyncratic responses to environmental gradients. And later, Diamond, we talking about the effect of competition and how this can drive checkboard distributions, or Tillman about different ways the species can get distributed through the sites. So these ideas correspond to patterns in the way that species are occur and change through these sites. Finally, we have the idea of nested subsets that said that communities with lower species richness are subsets of species of the higher species richness communities. And this have also implications, for example, for conservation, to understand which might be the species or the sets of species more vulnerable. This is just an example of how these idealized patterns transform into the sites by species matrix. This is, for example, the nested subset when we see that the sites with lower species richness, for example, this one, it's just a subset of the sites with a higher species richness. And with all these, several authors, especially Presley in 2010, have come up with ideas of how to use statistics to kind of differentiate the pattern of a that different meta communities show. And they have come up with three metrics, three indices of a meta community composition that can tell us to which of these idealized structuring patterns of a meta community it corresponds. So first we can analyze the coherence, or how ordered is the distribution of individual species through these sites. The matrix, it's very simple. It just counts, it's just organized communities along their composition and counts how many gaps in a species occurrence are there. So a completely coherent matrix will look like, for example, this one, that for each site, there are no gaps in the species present when we order them according to their species composition. So if we have a negative coherence, we will have a checkboard distribution pattern, the idealized community proposed by Jared Diamond, where species are competing and competition makes them like to be present or absent, but pairs of species to be present or absent. This was initially proposed for islands, but in general, we can think about, that checkboard pattern or showing high competition. We can have a random pattern which will show that there is no environmental gradient, or there is no organizing axis in a species composition. Basically, each species is behaving completely idiosyncratic in that, or it's occurring completely idiosyncratic in that landscape. And then if we have positive coherence, which shows that there are continuities in the distribution of a species through these sites, we can infer that there is a gradient, an environmental gradient, even if we don't know which one, that it's affecting or influencing a species occurring through this landscape in each of the local communities. Then we can assess turnover, or how much changes, how much boundaries of a species distribution occurs. And assessing turnover, we can know if there is a nested subset of a species, like the one I mentioned, in which a species at lower richness sites are subsets of the species at higher richness sites. Or if not, we can assess if the species are evenly spaced, or Clementian, in the sense that there are groups of a species that occur at different sites, or Gleasonian, in which each species like shows different responses to the environmental gradient. So with these three matrix, basically we can feed a data on species presence, absent, occurrent to one of those idealized patterns. And after that, we can try to infer what is driving that pattern and understand the mechanisms behind a species composition. So with this in mind, my objectives were first to describe a sampling completeness of the mammals for the Mongolian vertebrate project, explore very rapidly diversity patterns, and then find which of these idealized patterns best fit the data of Mongolian mammal communities. So for this, we used more than 3,000 museum record specimens that are deposited in the Southwestern Museum of Biology, and included 22 of the sample sites that had more than 50 samples. This to make sure that at least we have a more or less complete sample of the site, and with this we can estimate diversity and assess that we have a set of the majority species present there. Then to confirm this, we estimated a species richness with extrapolation curves and also a sampling completeness, which is like the percentage of the species estimated to be left for being detected using the package as iNext, which again uses species accumulation curves. As we get sampling units, as we collect individuals, how species diversity and sample coverage changes. Then using the package Metacom that uses the statistical and framework of Leibel, Michelson, and Presley, the one that I was mentioning you and showing you in the graph of the three metrics we can use to sample, well, to assess the patterns of the meta community structure. And using this package Metacom, I simulated different random communities, estimated the indexes from those communities, and compared what we found from the Mongolian, the observed communities with the expected from null models, and assess the significance of these estimated metrics. So we used 22 sample sites. The sample coverage there was from 85 to 100% of the species estimated to being detected. So this shows it was a very complete fieldwork, and that the analysis here are not biased because species were missed. And each site has from 50 to 484 samples. Sites had from 6 to 21 detected species. Here you can see the number or the species richness as a, represented by the size of the dot. Unfortunately, I think I didn't pick very well the colors, but you can see that in general, there are more species richness sites, or communities have more species richness towards Western Mongolia and towards the mountainous area of the country. Again, sample completeness was high, so we can be a little bit more confident about our results here. Here is a graph of the distribution of a species through the sites of Mongolia. Unfortunately, for space in the picture, I couldn't put here which site belonged to which, but here in general in the top are the Western sites. So you can see here the pattern that shows like how a species occur through the region in Mongolia. So I have a question for you. Looking at this pattern, which one do you think will be the idealized pattern for Mongolia? Any guesses from here?
So what's the question, Sebastian? I'm not sure of the question.
The Glissonian?
So,
based
we
on,
have
okay,
here,
now I see.
This is our data, right? Here it's the data,
yes.
how on the left, the sites, and on the top, the species. And so how species occur through the sites through the sample sites.
Okay,
right?
so then looking back at the different.
So these are the idealized ways and a species can occur through a landscape, right?
Yes.
Can you guess which one might be the most, like the best fit for it?
Well, I'd say that the evenly spaced one is my guess, but what does everybody else guess? I think
Glissonian.
Glissonian quasi.
Oh yeah, don't pay attention to these quasi nuances, but in general, it's
Sounds like a good name for a
So-
dog.
quasi.
Like on-
I also had, when I saw the graph, I also guessed it was evenly spaced.
Initially.
But then when we compare our results to random models, we can see first that there is coherence in the meta community, which means that the range of sites of occurrence of species are coherent with an environmental gradient. Even if we don't know which gradient is this, it shows that they are not some randomly occurring, but that we can organize the sites according to their species composition. So after that, we assessed a species turnover. So it's how many at each site, how many species range changes. And we found that it is significantly lower than what we would expect by random. And this means that there are not as many changes in boundaries as we would expect. So we have a nested subset of nested subsets of species. That is how it shows it, they changes. So sites with more species richness tend to be, or to have subsets of species from the higher richness sites. That's what this metric tells us. And finally, doing the third metric, which is clumping, or how often like the species appear or disappear together, we find that this is significant, and we have clumped species lost. So it's not idiosyncratic by species, but different groups of species disappear from the communities together. Of course, this is the idealization of the communities, right? There are lots of variation, and this is a continuum, but according to these metrics, there is a statistically significant trend towards these patterns in the community. So with this, we can conclude that the small mammal assemblage in the study area presents a nested pattern of meta-community structure, and the groups of species are lost along the environment gradient. So as the sites changes, groups of species disappear, and the more depauperate sites are results as subsets of the species that occur in more diverse sites. Any questions or comments so far? Clear as mud?
Very interesting. I think we could utilize this method to examine our grape data from years ago too. Very interesting.
That would be great.
Yeah, we could reanalyze that. I think we have it all on disk. Go ahead.
Okay, so after this, it comes the question of on what are the potential mechanisms driving the this pattern of meta-community composition. So for the first question will be, what is the gradient or the environmental gradient driving this, if there is one? And what are the effects of current and past environmental conditions? Finally, it will be interesting to see, like now that we know that species disappear in groups, like disappear from communities in groups, or never made it to communities in groups. It will be interesting to see if biogeographical barriers have something to do there, and what we are seeing is the effect of of dispersal limitations in certain groups of species. But these are just ideas. So far, what I did was to plot the order of the data, or like the sites were ordered according to their species composition, right? So the order of each site with the against its current and temperature during the Pleistocene, and found that there is a correlation, an important correlation of around 0.5 and 0.5 for current temperature and 0.6 for um, temperature during the last glacial maximum, suggesting that temperature is one of the factors driving this species composition of mammals. And it's very interesting that the past temperature was actually the one that showed more correlation with the ordination of the sites. So as again, this is preliminary data, but it will be interesting to formally assess which is the environmental variable, and if it's a current or past variable, the one that it's more related to this pattern. And then precipitation showed an even higher correlation with the ordination of communities for both current and past precipitation, suggesting that this is a very important variable in structuring communities, which makes sense with what we know with the ecology of the area, and knowing that parts of the study area are dry and might impose like ecological and physiological challenges on the species. Finally, the future work will include to assess if this clumping of species ranges match biogeographical or environmental barriers. It will be interesting to see also how phylogenetic diversity is distributed to the country, through the country, or through the study area, to assess if there are if these dispersal barriers or limitations is what is driving what the pattern we are seeing. And finally, it will be very interesting to do the same analysis for other vertebrate groups, such as birds, which is also data that was collected through this project. Finally, I have a couple of questions regarding parasites that I think I would like to know what you think about them. And it's, what about parasites? First, will you expect the same pattern, so that parasites communities are subsets of, well, yeah, parasites also showing a nested pattern where parasite communities with lower species richness are just a subset of the more diverse parasite communities through the landscape. If we saw this is happening with the parasites, what would you think with the hosts? What would you think will happen with the parasites? Any ideas?
Very good question. I think we need to look at the data and see what we come up with. We did some work with fleas. Do you have those flea data, Sebastian?
No.
Yeah, so we did, Gabor and I analyzed the flea data, so we do have some flea information, but we haven't done it for the endoparasites yet. There's still a lot of work to do because we haven't identified them all yet. So I think that we would see some of these things nested, especially the teniids that utilize mammals as intermediate hosts. So each one of those teniids that we see in this petri dish here are it's a tenia, and it's called Tenia polyacantha. Each one of those individual teniids came from an individual egg from a carnivore that was shedding eggs in its feces. And so these would have a different kind of distribution, a different distribution through space and time, because the intermediate hosts, they don't multiply in the intermediate host, they go in the intermediate host and then they're transported around. Some species of Tenia multiply in the intermediate host, and so we would have multiplication there. So we wouldn't need as many of them. Anyway, I think would be a really intriguing thing to look at both of these, and we could look at ecological fitting and host switching through space and time with these things, and integrated with your analysis would be really interesting to see. So,
Great.
yeah. But anybody else have any questions or comments? There, go ahead, Augie.
Also, we're thinking to add a network will be really cool because, very few diversity of the host in this case, but the parasites will involve some invertebrate intermediate hosts as well. Then
That's
you
true.
can figure out which one would have a more robust connection and responsible for the community assemblage. So that'll be something that we can look later.
Yeah, this would be cool too, because Pintinia polyacantha only depends on the small mammal contacting a fecal pellet of a fox or a wolf or some other carnivore that's infected with the tinea. So, so the model can be developed looking at the frequency of response of the small mammal interacting with the feces of carnivores. Anyway, I think it'd be pretty cool to look at, and the network, of course, would show that up to enable us to see those things. Go ahead, John.
So this may be a really dumb and ignorant question, but some of the small mammals that you've been dealing with have very wide distributions at the generic level. So Microtus is being a perfect example. And some of the others have evidently have a much narrower geographic distribution at the genus level. So I'm wondering what you might find if you just plucked out all of those mammals that have, which at the generic level are very widely distributed. So, and the kind of the ignorant person on the street question might be, do they bring into the mix some sort of phylogenetic level traits that allow them to do things that members of other genera don't have? That's just one of those wild ideas. What would these, go back to those R squared curves? Not this one. There you go. Yeah, that one and the precipitation one. What would those curves look like if you took out any of the mammals that have almost global or hemispheric distributions?
Yeah,
That's a very good.
Yeah, if we removed the Alticola, for instance, and then reanalyzed it with some of the more, so Alticola, let's see, there's stretch.
Alticola here.
Yeah, so that's Alticola there. So Alticola is really widely distributed, and there's also a couple of Microtus that are widely distributed there too. So that would be an interesting thing to look at. I'm not sure if it would make too much difference because it looks like they're not really contributing that much. But
right.
Spermophilus is one that's spread all over the place, but it's got a narrower distribution.
Microtus.
Microtus mariones, of course, that's gerbils.
Gerbils, yeah.
So cool. Yeah, the interesting question. We could like do a bootstrap or a bootstrap analysis of this and rerun it over and over, removing and replacing to see what would happen.
Yeah, we can do it, and I think that will be the best way to see. My guess is that if we remove the most widespread species, we will lose the nested subsets pattern because, well, I will guess that the most depauperate communities, well, the ones with the lowest species richness, will be composed mostly by the wide and very widely distributed and more tolerant species or groups. But yeah, that will be a thing we can do.
Well, you might pick up, you might pick up the effects of a barrier of some kind if, by taking out and again, this is just naivete, but you'd think that a widespread species, or at least members of the widespread genus might not be so impacted by a barrier that kind of an
Yeah.
evolved, narrowly distributed species might, even though there might be appropriate habitat, across a barrier somewhere.
Yeah. Yeah, that's a very good question, and we can like easily try just to remove it. Thanks.
Yep.
We can do that for the paper in Air Fortune.
Yeah.
All right, well, this river picture that you showed there is amazing. That's the Bulgan River that runs down in from the Altai down into China. We got there and I realized I didn't have my fishing rod. All I had was the fishing tackle and some line. So we tried dental floss with a couple of spinners, but we weren't successful. Anyway, so I'm excited about this this idea. Do you think that the dispersal barriers? I think the dispersal barriers might be easy to pick up once we do that kind of an analysis, because we can see that the dispersal barriers will consist of like desert steppe regions versus high altitude mountains, for instance. So
Yeah.
if we look at that one, I think I sent you one image today or last night that had the altitudinal gradients that we had used, that Gabor had analyzed for us a few
Yeah.
years ago. So that would be another thing to look at it simultaneously here.
Yeah, exactly. Like habitat associations.
Yes. Well, actually, it was altitude associations, which would have been, correlated to habitat. So cool. So, any other questions or comments on Sebastian's interesting talk? That thing's called a hopo. That bird, if anybody's interested. Hopo.
Hope.
Yeah, it's
Down
epops.
down in Oklahoma, they'd call it a hopo.
Yeah. So they occur all the way, I guess they're whole Arctic. So.
Well, they're sub-Saharan also, at least
Yeah.
the genus is.
Sebastian, I just wanted to ask, it's not really directly related to your work, but when Scott introduced you, said that you work for an NGO, and I was wondering, what is your NGO, and maybe does it have anything to do with your research?
Well, it's an NGO focused on biodiversity conservation. So we want to do field work, field work and research that has impact on biodiversity conservation. That's our work. And yeah, definitely, we want to include parasites in all of these and use DAMA protocol for these kinds of assessments. That's the main goal. Currently, we're doing a project on camera trapping of endangered species in an Andean region that has been not very well sampled. So we're trying to understand what is there, and we're also working with coffee producers to try to promote better practice, like promoting wildlife friendly practices by the producers and try to start standardizing these. That's like the project we're in now. But yeah, definitely we want to include this type of research and in our work.
Excellent. So what's the name of your NGO again, just for all of us?
It's a Laboratorio de Conservación Colombia, like Conservation Laboratory Colombia.
Nice, good name. Any other comments or questions from anyone? It's really great to have you back with us today from Medellin. It's really cool. Thank you so much.
Sebastian.
It's really great. We miss you a lot.
Great to see you.
Yeah,
everybody. Yeah, I miss you guys.
Yeah, it's good to see everybody in here too. And we have people from all over. It's really exciting. What a wonderful seminar. So this is the last of the seminars for the semester. Let's look at your last slide there, Sebastian, before we end here.
Oh, no, well, thank you.
Oh, yeah,
first to everybody who collected the data. I wish I could have been there, but
Yeah,
to everybody that collected the data, and thanks to all of you for listening to me.
yeah, this shows all the way from 1999 up to the last year we were there, 2012. So what a group. What an amazing operation we had. So thanks a lot. So,
Okay.
all right, well, thanks a lot. And we will be back here starting in August. I'll send out email and Facebook announcements. You're going to get a bunch of Facebook announcements about our field parasitology class as we're teaching it this summer. We're going to go ahead and teach it live at Cedar Point. There will be some people who are going to take it remotely because of their needs. And so we will be live and remote, and it will be quite exciting. Augie will be the TA, and we're going to be training Kevin out there. So anybody else wants to join us, please feel free to come in anytime on our feeds that we have running. We'll be using Discord for the feeds for the most part. So anything else for the group before I go ahead and stop? And I'm going to go ahead and turn this over. Once we stop, the recording will come to me, and I will put it onto our Facebook page. So I appreciate everybody being with us this semester, and good to see everybody on. So hasta pronto, hasta luego.
Hasta luego. Hasta
Adios. Have a great
Adios.
summer, everybody.
Yes, and please keep in touch. We're pretty easy to keep in touch with these computers. So I don't want to hang up, so I'll stay on here until everybody else leaves, I guess. This picture of Botero-Canola was from 1999. It's when we were just kids. That when he's sitting in the gauze. So
And look at Augie. How old were you there, Augie?
I think it was 14.
20, 26, 25.
Yeah, that was 2009, wasn't it?
Yeah.
Yeah. So Jean-Pierre. All right.
Manter, Yates.
Yeah. All right. Anything else from anyone? It's I hope everybody's getting their vaccinations going along. I know that most of us have. Some of us haven't. My mom got hers finally. I know that Griselda down there in Mexico hasn't gotten hers yet, but apparently she's scheduled, and Scott's got his. So we everybody's moving forward with that. So hopefully we'll get a bunch of people immune, and it will stop this thing so we don't have a lot more mutation going on. So all right, well, take it easy.
Okay, goodbye.
Yeah, so Sebastian, let's go ahead and put this, maybe we can put this down into a paper, get this thing finished.
Yeah.
So we can maybe start working on it next week. What's your schedule like next week?
Let's do it. Yeah.
Okay, so
Perfect.
let's try to figure out